Well, thanks very much, Pat, for the very generous invitation to join this meeting. I'm both humbled and honored to be invited to speak at this meeting. I was thinking back, the last time I was in a large gathering like this was actually in January 2020 in San Diego, which was a PPMD meeting also. So for me, it's coming full circle in terms of public events. So I'm very glad to be here to talk to you today. My remit really is to reflect on some of our experience with clinical trials in Duchenne and some of the learnings we've gained from working with you, both in the clinical trials, but also with some other projects, and all of the benefits that have accrued from doing that. So just for background, I head up rare disease clinical research at Pfizer. I work in the rare disease research unit for Seng Chen, who might be here somewhere, my boss. And we work to identify new therapeutic possibilities in many indications and bring them out of the lab into the clinic and to run early phase clinical trials to get to a point where we can be confident that they're doing what we want them to do. And then we can transfer them to our late phase colleagues who look after the larger phase 3 programs. Next technical problem, right, there we go. So, of course, I'm an employee of Pfizer. I own stock, and these views that I'll be expressing are not necessarily Pfizer views. There are many personal opinions in there. I wanted to just show our values and behaviors really as a prompt for me to talk about a culture shift that has been occurring at Pfizer over the last few years, partly driven by our new Chief Executive Officer, Albert Bourla. And I don't normally like these sorts of statements. I'm a bit of a skeptic. But I really like these ones. And I'm reminded of the behavioral recommendations when I joined Pfizer, one of which sticks in my mind, which was no jerks. So this is a real step forward culturally. But I mention Albert Bourla not casually, because he's transformed the vision of the company, given us courage to start to think about higher risk propositions through the work that he did to drive the company to deliver vaccines and antivirals in impossible timescales. He says hi, by the way. I don't say this because I'm a great mate of him, but I happened to meet with him face-to-face, one-to-one yesterday. And I told him I was coming here, and he wanted to share the greeting. And I know that he's very supportive of our DMD work, of our programs. And he shares my deep respect for this community and what you've been able to achieve over the last 10, 15 years. So you know all about PPMD's activities in terms of funding research, of advocating for, with legislators, of advocating to institutions, to companies, and of all the impact that you've had on improving care by sharing best practices and the Certified Center program, of the activities around your community in building social support as exemplified by this meeting. But probably what you're a little less aware of is the work that goes on behind the scenes, supporting and encouraging companies in small ways that add up to having a big impact on us and our ability to prosecute clinical trials and to actually transform our own cultures. And I'm hoping that through a few examples, I'll be able to show you a little bit about how that works. So I'm going to talk to you about the domagrozumab programs and some of our lessons from it, but also some lessons we learned from other collaborations with PPMD, and talk a little bit about the facilitation and guidance that you've provided to help us with our gene therapy programs. And if there's time, there probably won't be, I would like to talk about next generation therapies, which is a particular passion of mine. But I'm aware that there's plenty of other discussions of that type, so we'll probably not get to that. So in 2012, we just completed a phase 1 program looking at our anti-myostatin monoclonal antibody in human volunteers, healthy individuals. And we wanted to move this forward into the clinic. We held a medical advisory board. We got a lot of conflicting advice about how to design a clinical trial. At the time, there had been some recent disappointments in the field. You know, people had different views about how to move things forward. And we made some fairly radical proposals, we thought, to many individuals, including PPMD. And for the first time at Pfizer, we took an early draft protocol to some of your patient and parent panels to get input into some of the detail of the protocol related to the use of placebo and the acceptability of intravenous delivery on a monthly basis and some of our inclusion-exclusion criteria. We had discussions around endpoints and how to look at how to support families who are having to travel long distances to sites and so on. We talked about the burden of MRI scanning and how we might reduce that burden so that we could get information from muscle scans in a global clinical trial. And Pat said to us, oh, have you ever thought of including a patient representative on your data monitoring committee? And that was something we had never done at Pfizer. She happened to mention that PPMD had been training people to perform such functions. And so we did indeed include a representative of this community onto our data monitoring committee. And it's been a great success, and we've carried on that tradition in a number of our rare disease programs. So a small suggestion that's actually changed how we do our drug development. The Drug Development Roundtable was a great forum to introduce us, who are neophytes to Duchenne drug development, where companies were able to share views and share best practices. And that's been a very helpful organization over many years. So what we eventually came to was quite an ambitious study where we're trying to compress development, really compressing phase 2 and potentially phase 3 studies into one, which we managed to get the regulators to agree, if we were successful, that this would be a registrable trial. All quite radical in Pfizer, in the Pfizer world, and fairly controversial outside of Pfizer. One of the controversial areas was our selection of the 4-stair climb as our primary endpoint. And I'm kind of glad that Craig isn't here live in the next session, because he always gave me a hard time about that decision. But it was based on reasonable science that the 4-stair climb endpoint was very able to detect improvements with steroids, and actually the statistical performance in our hands did seem to be rather better than some of the other endpoints. Anyway, we recruited over a couple of years 120 boys into this study. They mostly got increasing doses of our antibody that we had good evidence in animal models would do some good in increasing muscle volume and strength. A huge amount of effort at 30 sites globally, a large internal Pfizer team that put their heart and soul into it. I'll sing the praises of Shannon Marafino and Tara McDonnell, Lawrence Charnas, who worked very hard on this study, and Catherine Beaverson, who did enormous amounts of work to make this feasible. So they were all disappointed, and of course, the families of those 120 boys were incredibly disappointed when we got to the point of looking at the data and realizing that we had not achieved our primary endpoint. The drug was safe, which was great, and we have a really useful safety database now of a clinical trial in Duchenne that we've used in many ways and is available publicly through the DRISC consortium. And then we looked at some of the other endpoints just to make sure that we hadn't missed something using the forced air climb. And we saw no statistically meaningful improvements in any other endpoint either. So it really did look as though this drug was doing nothing. And the top graph there, you can see this is really a flat line of showing the lack of response to the drug over a 12-month period. But it was perhaps the first lesson that we got from this study was that there was a flicker of activity in the North Star ambulatory assessment. And we'd seen this in some other also negative trials previously, suggesting that perhaps it's a more sensitive indicator of change in this population. And we went on to partner with the CTAP organization to ensure that that endpoint was interrogated a lot more closely so that we understood the natural history of it and its operating characteristics much better. The second lesson that we got from this data set I wanted to flag was that we saw, much to our surprise, a very significant proportion of boys who had elevated cardiac troponin I. Now, what's that? It's a protein that's made only in the heart and is only really released into the circulation when heart muscle cells are injured or die. And we found 20% of boys in the age of 6 to 16 had high levels. They wouldn't stay high for long. They were transient, but we consistently saw a significant proportion of boys with elevated levels. It wasn't associated over a 1 and 2 year period with an impact on cardiac function or clinical symptoms. But because of the biology of this biomarker, it does suggest that, and reinforces the argument that we need to pay more attention to heart health even in the younger population, that there is subclinical transient cardiac injury occurring quite frequently and really needs to be an area of greater focus. And I'm delighted to hear about the investments in the natural history cardiac program. The other thing that we put quite a lot of effort into was the MRI scanning. And I think it's turned into a very valuable data set. I mean, clearly there was evidence emerging from natural history studies that some measurements made by MRI could be very useful in tracking disease progression. So we committed to including this in the study, and the boys in the study every 4 months for 2 years had MRIs. Sarah Sherlock, who was our clinical imaging expert, put huge effort into working on protocols to make the whole experience much less stressful. And we found that even in boys of the age of 6, we were able to conduct these scans without any sedation. And across the course of the study, 97% of the MRI scans were interpretable. It's not idle data collection. We did see in the measures trends towards differences between our active drug and placebo. It was only statistically significant when we looked at T2 mapping, which is a measure of muscle edema and inflammation and fat content, where we did see a statistically significant reduction. However, of course, it wasn't associated with clinical benefit. So we went on to look at those MRI measures to see how predictive they were of ultimate loss of ambulation over a 2-year period. And they certainly predicted quite well. Also, change in the MRI at 12 months was clearly correlated with change in function at 24. So it was a very useful experience and data set that we applied in the gene therapy program subsequently to study muscle by MRI, and we're working through data from that study that will be presented later this year at the myology meeting. Okay, so I wanted to spend a couple of minutes talking about some other collaborations that we have worked on with PPMD. One was the newborn screening pilot that Nikki has been leading, and a little bit more about a patient preference study that Ryan championed, and you've probably seen the results before. But I do want to emphasize what's been important to us in those data sets that were generated through collaboration, and the impact both scientifically and culturally on on Pfizer. So the newborn screening project was a PPMD initiative, and we got involved really just to add in-kind support from Catherine Beaverson, a genetic counselor, statisticians, biomarker experts that PPMD were able to call upon to help support this effort. And I am delighted to say it's moving forward very well into a submission imminently with the RUSP, which is the list that the Department of Health runs as a recommendation for inclusion in newborn screening. So we were glad to be able to help with this, and it marks a model that other companies can follow of lending in-kind support. It doesn't necessarily mean donation of dollars to the effort. Oops. Oh, no. Isn't it always the way? One back. It's just like my TV. And this is the gene transfer patient preference study that we talked about before any of the gene therapy clinical trials were starting. Pfizer and Solid worked with Parent Project Muscular Dystrophy and Ryan to pull together this proposal, which was conducted really to understand individual preferences in terms of benefit risk, because we were entering a time when we had no real conception of what the safety profile of high-dose AAV therapies was going to look like, and we wanted to get some impression of how individuals with disease or families with disease would think about some of the potential risks. So the first aim was to find out about priorities in terms of whether people would join a clinical trial, which was important to us as we were about to launch one. And the third aim that I'll talk about is the perception of risk of death, which we obviously all hoped would never be an issue for any of our gene therapy programs, but important to use these sort of quantitative approaches to get a real estimate of what people think about it. So approximately 300 adults with Duchenne and parents or guardians with Duchenne were interviewed with a background story of what we thought might be the profile of a successful gene therapy, and asked them to prioritize various potential benefits or limitations or risks to see what would be most important to people. And what surprised us a little was that, of course, the degree of benefit in terms of muscle function and cardiac function were higher in the priorities. But when it came down to it, people were less concerned about whether there was a placebo or whether the study involved muscle biopsies. And that understanding was helpful for us in proposing our clinical trials subsequently. The third piece asked individuals to imagine a hypothetical therapeutic benefit, which would be a gain of delaying decline or progression of disease over time. For that benefit, what risk of mortality would people accept and rank it in a certain way? And what was interesting here was that actually the degree of tolerance to the idea was somewhat less, people were a little less risk averse than we had anticipated. But also that it was a bit context dependent in terms of the stage of disease that people were thinking about and which population was answering the question. So, I mean, we learned quite a lot that was relevant to our studies and their design. It was clear that this kind of structured data from weighted assessments is very valuable sort of objective quantitative information to put in front of regulators and ethics committees, perhaps eventually payers. But it also stretched our internal knowledge and know-how of this type of social science, if you like, methodology. And we are planning other similar activities in other disease settings. So, our work with you has taught us how this should be done and encouraged us to apply the similar types of methodologies in other settings. Okay, I want to just switch gear then to talk a little bit about the gene therapy. This is the only thing I'm going to say. If you want to hear updates of the gene therapy programs from Pfizer, Cassia Lobello will be reporting on both phase 1b and the current phase 3 trial status tomorrow morning. So I'd encourage you to listen in there. But back in 2018, gene therapies offered a whole new hope with unknown risks before we were giving these high-dose vectors into boys for the first time. We knew all about the non-clinical safety profile, but it's certainly our view that in this kind of new field that it's really important for the whole field to learn about the levels of gene expression and clinical efficacy and the safety profiles as early as possible. Of course, with safety, we'd normally inform our principal investigators in the trial, and we'd inform our patients through some sort of informed consent process when there are new safety findings. But such was the level of interest in these programs that we felt it really important that the whole community was aware. And we struggle with this as companies sharing this kind of quite sensitive safety information in a public forum. And we had endless arguments internally about whether this was the right thing to do. But through discussion with Pat and other members of PPMD, we're encouraged to be very public about it and talked in short order about the safety events that occurred early in these trials. I think the consequence has been not only have we built trust between PPMD and ourselves, but also with the community. And it runs against the grain for commercial companies to do this quite this way. So it really requires an active process of saying we need to, and we owe it to this community who are putting themselves forward into clinical trials to be very clear about safety events that we've seen. And this experience with the DMD gene therapy program, I think has had knock-on effects, again, internally at Pfizer. And we're applying similar types of ethical reasoning to other rare disease programs. So the influence of PPMD goes very much beyond Duchenne itself. All of the learnings that we had gained in working with the community have now been implemented in the phase 3 program, and I thank Nicholas for this slide, which summarizes some of the activities involving patient advice and input into the phase 3 program that were performed in the development of that protocol and indeed continue in the conduct of the study with patient representation on our clinical steering committee and our external data monitoring committee. So those learnings have been adopted. How am I for time? I have no idea. Am I? Okay, I thought so. So I will skip that. So I really wanted to take the opportunity to congratulate PPMD and you for all of your incredible achievements over and above what could reasonably have been expected in laying the foundation for the current explosion of novel therapeutic developments, for your thoughtful and open but responsible communication practices that both informs us and informs the community and holds us to account. You know, it's not unknown for Pat to call us up and say, what on earth are you doing here? It's clear that open collaborations can raise the standards for all participants, and I think the activities where we have collaborated in active research have been great contributions to the literature that everybody can learn from. But fundamentally, we in companies have a great deal of expertise in some things, but we need guidance on how to apply it in a complex disease setting like Duchenne. And without the support and encouragement and input to our activities and processes that we get from PPMD and other like-minded organizations, we really wouldn't be able to move programs efficiently. So I thank PPMD and for all of you for that. I'm optimistic that through continued dialogue and innovation, we can prepare to meet all the future demands of boys and their families to solve many more of the problems, as we just heard for a small population from Kevin. Anyway, thank you again for the invitation, Pat. I'm very grateful and honored, and I hope you can see the degree of interaction that occurs with your organization and the impact that it's had not only on our technical processes, but also on our culture. So thank you.